Hello and welcome everyone. So, today we'll discuss yet another very interesting topic and a concept that is telomere and telomerase. So, let's begin. What is a telomere? A telomere is the end piece of chromosome, and so this end piece of chromosome is called a telomere. So, here are the telomeres of this chromosome. Now, what are these telomeres? They are repetitive sequences that code for no particular protein. So they do not have any genes in them and they are repetitive sequences. In human beings, these repetitive sequences have a 6 base pair sequence, that is TTAGG, and it is repeated 100 to 1000 times. These telomeres protect the important genes from being deleted as cells divide and as DNA strands shorten during replication. So, this is the function of the telomere. Now, what is telomere shortening is the next question. So, after each round of DNA replication, some telomeric sequences are lost at the 5' end of the newly synthesized strand on each daughter DNA. But because these are non-coding sequences, their loss does not adversely affect the cell. After sufficient round of replication, all the telomeric repeats are lost. If shortened beyond some critical length, the cells cannot survive. In a normal somatic cell, telomeres shorten with each successive cell division, and this is known as telomeric shortening. In germ cells, stem cells, and cancer cells, telomeres do not shorten. So the cells survive long. Now let us look at the aging and telomeres. So what connection does aging have with telomeres? You can see here two chromosomes. This is a younger chromosome and the telomeric regions are much longer, and the second chromosome is a older chromosome with short telomeric length. So it means that with successive cell divisions, with time, the telomeres undergo shortening, and this reflects in the aging process of a human being. So this is the aging process of a human being, which is correlated with telomere shortening. Now, the telomere shortening is also related to various age-related diseases as shown in this diagram, as referenced here, that almost every organ of our body is affected due to the telomere shortening as the age advances. The eyes, the brain, the lungs, the artery, the metabolic uh, diseases, the liver, heart, bone marrow, kidney, ovary and testes, the bones and the joints. So all these are affected and these are implicated in various diseases. Now we are faced with all these problems due to an end problem which is seen in a linear DNA replication. So let us look at what is the end problem of a DNA replication. As you all know that we are eukaryotes and the prokaryotes have a circular double-stranded DNA, whereas the eukaryotes have a linear double-stranded DNA. So because our DNA is linear, it has an end problem. So the linear chromosomes have an end problem, and in this the lagging strand utilizes various RNA primers to form the Okazaki fragments except the end part. So the last part of the lagging strand is unsynthesized or unreplicated, so the DNA polymerase enzyme is unable to synthesize a new strand at the 5' end of the uh, lagging strand. Now, about 300 nucleotides in the 5' end of the new strand cannot be replicated. So, after DNA replication, each newly synthesized DNA strand is shorter at its 5' end compared to the parental DNA strand. So the two daughter DNAs have 3' overhangs at opposite ends. Now telomeric DNA replication is, let us understand this better to understand the end problem of linear DNA. So this is a linear DNA, this is the leading strand, this is the lagging strand. The leading strand has a single RNA primer on which the DNA polymerase acts and synthesizes a new strand, whereas the lagging strand has multiple RNA primers and small fragments known as the Okazaki fragments are synthesized. So as the replication reaches its termination stage, the uh, primers have to be removed, and these primers are made of RNA. So now this RNA primer is replaced by DNA primer. Let us see how. So first the RNA primer is lifted up like a flap by flap endonuclease, one enzyme and RNase H. Once it is lifted up, it is cut and removed, and the RNA primer is replaced by DNA by delta polymerase enzyme, which is a eukaryotic polymerase enzyme. And uh, then these fragments are ligated. Now in the leading strand there is ligation, whereas in the lagging strand, since there are multiple Okazaki fragments, all the primers are removed, they are replaced by DNA fragments, and then these small Okazaki fragments are ligated. But at the telomeric end of the lagging strand, a small portion remains unreplicated. Now, why is this happening is because when the last primer of the Okazaki fragment is removed, there is very little space left for the DNA polymerase to to replace it with DNA fragment, and so that there is not sufficient DNA left in the leading strand, so the lagging strand cannot be synthesized, and therefore there is incomplete synthesis of the lagging strand, and this leads to, so after the final RNA primer is removed, there is no more DNA in the 5' direction. So DNA polymerase cannot replace the RNA with DNA. So both daughter strands have an incomplete 5' strand with 3' overhang. And this is how it is. So as this continues with successive cell divisions and replications, the length of the DNA keeps getting shortened. Now we come to telomeres and senescence. So what is happening is, as with there is successive cell division and as there is successive replication, with each round of replication there is some shortening, and uh, till the critical length of telomere shortening is achieved. So it takes around 70 divisions to reach that critical length, and once that critical length is reached, the cell division stops. So when the cell division stops, then the cell has reached senescence and aging, and since all the telomeric ends are non-coding, so the cell stops dividing and it doesn't want to harm the coding regions. And then there are various uh, age-related deteriorations, and which finally leads to cell death. So this is about telomeres and senescence. Now, telomerase is the answer to this telomere shortening, and various eukaryotic organisms have different ways of solving this telomere shortening problem. Human beings have a special enzyme known as the telomerase or the terminal transferase, which is abbreviated as TERT. And this telomerase enzyme has special features or abilities because of which it can prevent the telomere shortening in successive replications. So, what does this telomerase enzyme have? One, it has an RNA template, and second, it has a reverse transcriptase activity. Now, let us see how these two traits can enable telomerase to solve the problem. So, now let us look at the telomerase mechanism. Now, this is the end part of the newly synthesized DNA, which is not replicated. Let us see how the telomerase enzyme prevents this from remaining incomplete. So, the telomerase has an associated RNA that complements the 3' overhang at the end of the chromosomes. So this is the 3' overhang and this is the RNA template which complements it. So the telomerase comes and positions itself here. Then the RNA template is used to synthesize a complementary strand. So this is the RNA template and a complementary strand is synthesized. So the length of this leading strand increases. Now because of this, the telomerase moves forward and the telomerase shifts and the process is repeated. So there is further increase in the length of the leading strand. So because of this increase in length of leading strand, the eukaryotic DNA polymerase will now have sufficient space to complete the replication of the lagging strand. So what will happen is that the primase will synthesize a primer and DNA polymerase will now comfortably synthesize the complementary strand which it was unable to do earlier. So that is how the telomerase is enabling the completion of the terminal part of the lagging strand. Thank you. Like, comment, share and subscribe to my channel. Thanks for watching.